Hey, Mr. P here. In this video, we're going to talk about units of measure. We're going to focus on the metric system and unit conversion as a whole, but we're going to highlight the three most important units when we're talking about microbiology and microbes. Those three units being a millimeter, micrometer, and nanometer. Most of everything that kind of fits into the genre of science that is microbiology will fit into those three units. And you have to be really confident and accurate when you're converting between those three. In this class, we'll talk about unit conversion, we'll talk about size of microbes, we'll talk about calculation of magnification, we'll talk about calculation of actual size of microbes, we'll try to do some measuring with a microscope. And so it's really important that we understand those values or those units and how to convert between them. So when we think about units of measure, obviously it's a science class. We're going to focus on the metric, or we're going to stay within the metric system. And so the metric units that I'm sure you've all been, accustomed to seeing or accustomed to working with in your science courses, obviously starts at the base. And again, we're using this particular unit, which is distance. Okay, so we're going to start with our base, which is the meter. And then when you go down by a factor of 10, you have the decimeter. You go down by another factor of 10, you have the centimeter. You go down by another factor of 10, and you have the millimeter. These three, okay, or four, from meter to deci, centi, and milli, those are ones that you've been converting between for a long time. For the purposes of this class, though, we're going to focus on kind of this range, the micrometer, the nanometer, and then the sometimes picometer. We get into the picometers when we kind of get into, viruses or viral components. But for the most part, millimeter, micrometer, nanometer, those are the three that we're going to see a lot of in terms of units of the organisms that we're looking at in class. So how do we work on this or use these to help us measure microbes? Remember the order: millimeter, micrometer, nanometer. And unlike the fact that meter, decimeter, centimeter, and millimeter go by basically a power of 10, when you get to micrometer, you will see that it actually goes by a power of 1,000. Okay, so you have 3 extra zeros. So 1 millimeter becomes 1,000 micrometers, and 1,000 micrometers become 1,000,000 nanometers. Okay, so you basically add 3 zeros or 3 place values between millimeter to micrometer to nanometer. Okay, that's just the biggest difference. If we drew a line here, we would say everything above that line you're going to add a single value or a single zero when you convert. Everything below the line you're going to start doing a power of 3, okay, or a power of 3 place values, a power of 1,000. All right. So if you have a microbe that is 2 micrometers in diameter, that would actually be equivalent to 2,000 micrometers, and that would be equivalent to 2,000,000 nanometers. Okay. Now, obviously we're using in this example just, even numbers. We're talking about 1 in this case, or we're talking about 2 in this case, and so it's easy to see. You just add 3 zeros. But make sure you just understand that it is a power of 1,000, 3 place values to get from millimeter to micrometer, and 1,000 or 3 place values to get from micrometer to nanometer. This little image or this graph diagram will show you kind of what belongs within the range, or what the ranges of particular microscopes. Now, in future videos, we're going to talk about each of the microscopes in depth. We're going to talk about all of these bacteria. We're going to talk about the eukaryotic, prokaryotic, and even some viral components in future videos as well. But for the purposes of just this video, I wanted you to kind of get an idea of what size these organisms are, and how detailed, or how each of the microscopes kind of dives down into certain ranges while we're talking about micrometers and nanometers and etc. So the unaided eye will see and be able to resolve images that are down to about 200 micrometers. That is basically 2,000ths of a millimeter. Okay, that gets very, very small. light microscopes, okay, will go from about 1 centimeter to about 100 nanometers. Okay. Scanning electron microscopes, they'll go all the way down to about 10 to 15 nanometers. Transmission electron microscopes will go clear to the 10 picometers. And then you have some other microscopes which we're not going to talk about. But you can see that our use of technology will allow us to kind of extend our vision from that 100 micrometer scale or size limitation all the way into or through the smaller micrometer, the entire nanometer range, and then into the picometers. So I'm not going to go into the intricacies of each of these microscopes, but I do want you to kind of have a mental image of what these units mean. When we're talking about whole organisms, a lot of times you're actually above the level of millimeter. Okay. If you're talking eukaryotic cells, you're typically in the, upper micrometer sizes. If you're talking prokaryotic cells, you're in the lower micrometer sizes. If you're talking viruses, you're definitely in the nanometer range. And if you're talking about cellular or viral components like the DNA double helix, you're in the smaller nanometer range, if not into the picometers, depending on how big that particular cellular structure is. So that's all I got for this one. Make sure you are versed on unit conversion. Make sure you know kind of the order of the units, and I look forward to future videos. Thanks. Bye.